All right, I think we'll go ahead and get started. So good afternoon or good morning, everyone, depending on your geographical location. My name is Ram Dasgupta. I'm a senior group leader at the Genome Institute of Singapore, and it's my absolute pleasure to serve as a host today for this third installment of the France-Singapore Science and Innovation Lecture Series. So this initiative resulted from the recognition that bilateral cooperation between France and Singapore must continue to be strengthened, especially in the priority areas such as AI, infectious disease, economy, and innovation. And consequently, this is really a collaboration between the French Embassy in Singapore, the Collège de France, and the NRF, the National Research Foundation in Singapore, which launched the first of this lecture series in 2019. And this is essentially a continuation of that effort. So let's get started with the program. It is my honor to introduce the French ambassador to Singapore, His Excellency, Mr. Marc Avançon. Mr. Avançon is a career diplomat serving multiple portfolios, including Inspector of the Ministry of Foreign Affairs and the Diplomatic Advisor to the Ministry of Defense. And earlier in his career, the first Secretary of the French Embassy in China and subsequently in the United States. He was appointed the Ambassador of France to the Republic of Singapore in November of 2016. His professional experience mainly covers Asian affairs. In fact, he was an exchange student in the Department of East Asian Studies at Harvard. In the mid-90s, Marc is a former student of the École Normale Supérieure, from which he graduated as Associate Professor in Philosophy. So without any further ado, I would like to welcome Mr. Avançon to give the opening remarks for this conference. Marc.
Thank you very much, and thank you for your kind words. Professor Patrick Tan, Monsieur le Professeur Hugues de Thé, ladies and gentlemen, bonjour. I'm delighted to open this inaugural lecture of the France-Singapore Science and Innovation Lecture Series 2020-2021. This lecture series was launched as a result of the France-Singapore Year of Innovation in 2018 and 2019. And it's presented jointly by the French Embassy in Singapore and the Collège de France and the National Research Foundation here in Singapore. This lecture series started in 2019 and consists of presentations from eminent professors from France in Singapore and from Singapore to France. Hence, we had the honor to welcome in Singapore last year Professor Philippe Sansonetti, a worldwide renowned researcher in microbiology and infectious disease, whose work has been particularly valuable in gaining a better understanding of the crisis we are all confronted to. Early January, Professor Alain Fischer, Chair of Experimental Medicine at Collège de France, shared also about his work on curative approaches of gene therapy. 3 other professors of Collège de France will participate to this second series in the coming month. Professor Anne Chung, Chair of Intellectual History of China in February 2021. Professor Thomas Lecuy, Chair of Dynamics of Living Systems in April 2021, and Professor Pierre-Michel Menger, Chair of Sociology of Creative Work in June 2021. So I would like now to express my deep gratitude to our speaker today, Professor Hugues de Thé, who accepted to give this lecture online. And we hope very much to have the opportunity in a not too far future to welcome you in Singapore. I would also like to express my thanks to Professor Patrick Tan for welcoming this lecture at the Genome Institute of Singapore. And as you said, I mean, this event today is part of the 2020 edition of Voila, France-Singapore Festival, which is still going on till November 22nd. And this year, the festival gathers 50 programs covering culture, education, science, and lifestyle, and is placed under the symbolism of resilience, innovation, And it's also an opportunity for us, to experiment new formats for events. And lastly, last but not least, to reaffirm also the friendship between our two countries. As you may know, we are commemorating the 55th anniversary of the diplomatic ties between France and Singapore, and France was among the first countries to recognize Singapore in 1965. So without further ado, I invite Professor Hugues de Thé and to take the floor, and I wish you a very enjoyable lecture and fruitful discussion. Thank you very much.
Ram, I think you're on mute.
Oh, great. Can you hear me? All right.
Yes.
Thank you. Thank you, Mr. Ambassador. I would like to now call upon Professor Patrick Tan, our fearless leader and Executive Director of the Genome Institute of Singapore and Professor at the Duke NUS Medical School, to deliver his opening address for this lecture series. Patrick, as most of you are aware of, is leading the precision medicine efforts in Singapore through PRISM and directs the Sing Health Duke NUS Institute of Precision Medicine as well. He was trained as an undergrad at Harvard and received his MD PhD from Stanford, and he's made some fundamental contributions towards a mechanistic understanding of gastric cancers, of course, internationally recognized by numerous high impact papers, awards, and grants. And as he has an elected member of the American Society of Clinical Investigation, as well as a board member of the International Gastric Cancer Association. So with that, Patrick, the mic is all yours.
Thank you very much, Ram. And Mr. Ambassador, Professor De Thé, it's been a great honor, and on behalf of the Genome Institute of Singapore, to host your lecture on behalf of France and Singapore. What I would like to do in the next few minutes is to maybe talk a little bit, very briefly, about the Genome Institute of Singapore and the importance that international connections between countries like Singapore and France require in order to make advances, particularly in the field of cancer research and oncology. So we are very happy to host Professor De Thé's lecture today as the Chair of Cellular and Molecular Oncology. Professor De Thé's work in the understanding the molecular genetics of leukemia is extremely well known, as well as his ability to move between biology and medicine. But the work that he does is very much in tandem with the ethos of the Genome Institute of Singapore, which is to use genomic sciences to improve human health and prosperity. The Genome Institute of Singapore started in the 2000s. We are about 20 years old, so not so young anymore. And we have 4 major divisions of research: precision medicine, epigenetics, single cell systems, as well as genome architecture and design. We feel that the Genome Institute of Singapore is a very strategic choice for hosting Professor De Thé's lecture today, because his particular field of interest, which is molecular oncology, is also one of the key research focus areas of the Genome Institute. We are very interested in identifying genetic variants related to cancer susceptibility, and we participate in many national consortia related to cancer research, including national-scale liquid biopsy programs. We have expertise in different cancers that are endemic to Asia, such as gastric cancer, lung cancer, and also liver cancer, and also highlight Professor Ram Dasgupta's recent paper a few weeks ago in Cell, showing the single-cell analysis of liver cancer. So congratulations to Ram. We do believe that by working together, we can improve outcomes for cancer patients by making the right medicine to the right patient. And I think that Professor De Thé's lecture today will be a magnificent showcase of precision oncology. I also want to highlight that while we are in Singapore, we have many different international scientists at the Institute, in fact, several from France. We collaborate with the University Hospital on Angers, France in ophthalmology. We work with France Genomic on environmental metagenomics. And the GIS is home to several colleagues from France. For example, Nick Bertin and Maxime Hebrand, who are part of our computational biology group. We have PhD students, and we hope that once COVID lifts, many of the participants from France will consider coming to Singapore to spend some period with us in research, either for a short stint or perhaps for longer. So without further ado, I would like to welcome Professor de Thé virtually to Singapore, and I'll hand the podium back to Ram and today to start today's lecture. Thank you very much.
Thank you, Patrick. It is now indeed my great honor to introduce today's speaker, Professor de Thé. Professor de Thé is a professor of molecular oncology at the Collège de France and a physician at the St. Louis Hospital in Paris. He is a MD PhD physician scientist, receiving dual training in medicine and basic sciences, and in fact has been at the forefront of translation leukemia research. In fact, his research is very close to my heart, aimed at linking molecular cell biology to therapeutic responses in cancer. In fact, his classic studies on acute promyelocytic leukemia and the targeting of the PML retinoic acid receptor alpha by retinoic acid and arsenic has fundamentally changed the clinical paradigm, leading to the cure of this once what was thought to be an intractable disease. Needless to say, these studies have earned him significant international recognition. He's a recipient of numerous awards and grants. Professor de Thé is currently working on improving the therapeutic response on other leukemia through mechanistic elucidation of drug action in vivo. So with that, I would like to welcome Professor de Thé to deliver his lecture titled Dissecting the Mode of Action of Targeted Leukemia Therapies. Professor de Thé, the stage or this virtual platform is all yours. Really looking forward to your talk. And just a quick reminder to the participants, please use the Q&A chat box at the bottom to ask your questions, which Professor de Thé will take at the end of his talk. Thank you so much, Professor de Thé.
Thank you, Ram. Let me check this. Okay. Well, it's a great pleasure and honor to give this lecture in Singapore, even virtually. And I very much hope too that once the sanitary situation improves, I will have a chance to visit the Genome Institute, a place which I would very much like to visit. I would also like to express my gratitude to His Excellency, the French Ambassador to Singapore, Marc Abinsour, to the National Research Foundation, as well as to the Genome Institute of Singapore and its director, Patrick Tam. So without further ado, I will give you a personal view of cancer research, which has really been driving my work for the past 30 years. And I will not talk so much about technological driven cancer research, such as omics, deep sequencing, single cell genomics, artificial intelligence, functional genomics, organoids, metabolism, because I think that my colleagues in Singapore are probably much more educated than I am in this respect. These are developed in centers with very strong industry connections, and broadly the results are correlated to the investments. However, what we have learned from the past 20 years is that, targeted therapies are not necessarily curative, and that we need more than identifying a target and a drug to cure cancer. What we need really is actually understanding the mechanisms, and the classic example of Gleevec or imatinib is summarized here with two pictures, one from Time saying, you know, here we have the magic bullet that will cure all cancer. And almost 20 years later, a review by one of the key actors in the BCR-ABL Sun story, Brian Drucker, showing that we actually have response, but that these are transient, and we have acquisition of resistance, so that we really have to elaborate a strategy to overcome these resistance and to get cancer eradication. And so I'd like to make the case that most paradigm-changing discoveries were made by chance. and not so many examples of a priori designed breakthroughs. And just to remind you that history is often rewritten, because even Gleevec was not initially designed as an ABL inhibitor. And what I'd like to do today is to share a personal perspective based on 30 years of translational and basic cancer research, showing that curiosity-driven, unexpected observations starting from patients can also be a way to get into cancer biology. And naturally, the fact that we start from clinical observations will ensure ultimately clinical relevance. I'd like also to stress that actual cellular and molecular mechanisms for therapy response are important. And I'd like to argue that there are not many examples where we understand the mechanisms in depth and where those mechanisms are linked to clinical efficacy. And I'd like to make a bet that at least some of the future breakthroughs will rely on this type of approach, starting from unexpected clinical observation, constructing cellular and molecular biology from these observations, and then getting back to clinic. So what I'm going to talk about today is acute promyelocytic leukemia, a disease that was described in the late 50s by a Norwegian clinician, described to as the most vicious, the most fatal of all leukemias, because in those days, most patients would die a couple of weeks only after diagnosis was made. The first breakthrough was made by an American cytologist, Janet Rowley, who described a recurrent chromosomal translocation between chromosome 15 and 17, which was the molecular hallmark of this disease and led to the definition of this disease by cytogenetic means. And finally, the last element was the observation by clinicians in the US that retinoic acid, a hormone that was at the time investigated because of its activity on many embryonic stem cells, and notably the F9 or the HL60 model, could trigger terminal differentiation of primary APL cells ex vivo. And this was the driving force for the experimentation in patients by Wang Zhenghe and his colleagues in Shanghai, and of the effect of retinoic acid directly in patients, largely because in those days the Chinese clinicians had no other opportunity, no other choice. And he was able to make a remarkable contribution demonstrating that retinoic acid could induce clinical remissions, leading to from shifting the bone marrow from a purely leukemic state to a normal state. And this led us in cooperation with clinicians in the Saint Louis Hospital in Paris, notably Laurent de Gauss, to demonstrate that the retinoic acid receptor gene was a target of the 15-17 translocation. So what does this look like morphologically? Well, morphologically, this is a representation of leukemic cells prior to diagnosis. And this is what happens after 2 weeks of retinoic acid. All these cells are still leukemic in origin, but as you can see, they are differentiating towards granulocytes, and granulocytes are extremely short-lived cells, and these short-lived cells will be cleared away so that after a few more weeks, the bone marrow will be replaced by normal cells. So this looked like a miracle. It was promoted as differentiation therapy. The bad news is that these responses are often transient, most often transient, and that with very rare exceptions, all those patients relapsed. And so in Saint Louis Hospital, Laurent de Gauss initiated a few years later a combined retinoic acid anthracycline therapy, which induced cure of a substantial number of patients. And so, as I was mentioning, this led to the molecular identification of the molecular hallmark of APL, the 15-17 translocation. wherein through this chromosomal translocation, a fusion protein will be generated between the retinoic acid receptor alpha gene and a gene that we name PML for promyelocytic leukemia, creating a PML-RER fusion protein, which is really the main driver of transformation in vivo or ex vivo. So retinoic acid receptor alpha is a retinoic acid responsive nuclear receptor. So it's a transcription factor that which can be switched on and off with retinoic acid. And PML is, as we'll see, the organizer of nuclear domains, which are controlling many aspects, including senescence, at least in part through p53 control. And naturally, retinoic acid will bind PML-RER through the ligand binding domain of the RER moiety of the fusion, so that historically, retinoic acid activity on PML-RER positive APL was probably the first example of targeted therapy. So the first model which was proposed and which is still in many textbooks is the following, is that PML-RER binds DNA together with a poorly characterized transcriptional co-repressor complex and silences genes induced in differentiation. And so this transcriptional silencing is responsible for the differentiation arrest. And in the presence of retinoic acid, the binding of retinoic acid to PML-RER will transform this repressor into a potent activator, and the activation of differentiation genes will induce differentiation of the leukemic cells towards granulocytes, which are cells that will be naturally eliminated. So this really became a textbook model for transcriptional-based differentiation therapy, and everyone thought that the story was closed. Except that there was the comeback of Peter Ehrlich's magic bullet, arsenic, which he developed for infectious disease, and which was also developed in China in Huijing Hospital by Zhu Qian and Wang Zhenghe. And what they could find in relapsed patients who had relapsed from retinoic acid resistance is that there was clinical response at very high efficiency, over 95% complete remissions. And as you can see on this image, the morphological aspects of arsenic-treated APL patients, this is a bone marrow sample taken maybe 10 days after arsenic therapy. And as you can see, the images are essentially undistinguishable from those of patients treated with retinoic acid. And this differentiation in vivo is accompanied, in fact, in contrast to retinoic acid, by a very high curative efficiency, which from clinical studies performed in India and in Iran was actually estimated to be a 70% definitive cure using arsenic as a single agent without retinoic acid and without chemotherapy. And so the reason why we established a very tight collaboration with my friend Chen Su in Shanghai was that it immediately became apparent to us that this was completely incompatible with the transcription-based model, because arsenic does not do anything onto retinoic acid signaling. And so that this simple model of transcriptional reactivation by retinoic acid leading to differentiation was at best insufficient to explain its activity, and totally impossible to explain the clinical efficacy of arsenic. And so this led my group and a number of others to refocus on PML, which was really the constant partner of the fusion on which very little was known at the time. And as you can see on this image taken from a review, is that PML can form those bodies, those nuclear structures, which we envision as shells for post-translational modification of proteins. And by proteins, I mean a very large number of partner proteins, which are represented here, some very well-known, such as P53, SUMO, DAX, SP100, will be recruited by PML into these bodies. And subsequent work from my lab has shown that in these bodies, they will undergo post-translational modification, and particularly sumo conjugation, sometimes followed by degradation. And so what was discovered, and this is really a historical slide, is that PML-RER has the ability to deorganize PML bodies from these empty shell, which you can see here by immunoelectron microscopy or superillumination microscopy, where PML will concentrate those partner proteins. And physiologically, it was shown by the group of Pellicci in Milan that this is key in controlling p53 activation and senescence. And PML-RER disrupts PML nuclear body. This is an image of a transfecting cell. These are normal PML bodies in non-transfected cells. And when you transfect PML-RER, you have this microspeckled pattern, which we propose could be used for diagnosis, and it actually is in many countries. And this was shown by Pier Giuseppe Pellicci to blunt p53 signaling and to induce apoptosis and senescence resistance. And remarkably, we're able to show that these are primary APL blasts taken from a patient treated in Saint-Louis. And when you treat the patient with either retinoic acid or arsenic, these cells, which are still leukemic cells, will undergo this reformation of PML nuclear bodies. And this reformation of PML nuclear bodies is in fact the reflection of PML-RER degradation by retinoic acid or by arsenic. Thus, in contrast to the model of transcriptional control, which was only valid for retinoic acid, degradation of PML-RER by arsenic or by retinoic acid was the only shared feature between those two clinically active drugs. And so we made the bet with my friend Chen Su that there was a role of PML-RER degradation in response, and that reformation of PML nuclear bodies was likely contributing to the clinical efficacy of these two drugs. You have to understand that 25 years ago, this was a very obscene proposal, and that everyone was so persuaded that transcriptional control was key to biology that proteolysis, arguing that drug-induced proteolysis could have any clinical activity, was not very well received by the community. And so we took the efforts of going into the biochemistry of this degradation to understand it. This was done by Valérie Lallemand Breitenbach in my lab. And to make a long story short, retinoic acid will bind to its hormone binding pocket in the RER part of PML-RER, triggering reorganization of the by allosteric reorganization of protein structure, recruiting the proteasome and driving proteasome-mediated degradation of RER alpha or PML-RER. This is a general mechanism, a general feedback mechanism, which is shared for all nuclear receptors, probably to ensure cessation of transcriptional activation. And this was discovered by Jun Su in the lab. And binding of arsenic to PML on the site that we identified here in one of the zinc fingers triggers a complex series of events, which is just summarized here, where we have oxidation of PML through direct arsenic binding, polymerization of PML and nuclear body aggregation to create this external shell of PML nuclear bodies, which drives then the conjugation of PML by the sumo ubiquitin-like peptide, and then polysumolation of PML or PML-RER will drive recruitment of an enzyme which is a sumo-dependent ubiquitin ligase called RNF4, which will trigger polyubiquitination and recruitment of the proteasome and degradation by the proteasome. So it's very remarkable that those two drugs that were found by chance through clinical empirism actually have the shared property of binding directly to PML-RER and triggering a set of biochemical events that will result in proteasome-mediated degradation of the fusion. And so I think it should be clear from this presentation that arsenic also targets the normal PML protein. And you can see here this normal PML protein, and note here that you have a substantial amount of the protein which is present diffusely in the nucleus. And this diffuse nuclear fraction of PML will be recruited onto these very large nuclear bodies very rapidly, literally within minutes after arsenic exposure, and then will be followed by PML degradation onto these bodies. Okay, so there was a very long contention in the APL, in the leukemia world, of whether transcriptional activation or degradation were essential. And as often, it's the use of mouse models that was able to decipher between those two possibilities. And the first observation was that we could have genetic uncoupling of differentiation and leukemia clearance. And this was made by comparing in animal models PML-RER-driven APL. This is the standard classic APL, 97% of APL cases, and very rare cases of APL, which are called atypical variant APLs, where the fusion partner of RER alpha is another gene called PLZF. And PLZF-RER generates a leukemia, which is known to be insensitive, resistant to retinoic acid. And in these mouse models, what we're able to show is that after retinoic acid treatment of the animal, we have a first phase of differentiation and then replacement by the normal bone marrow as soon as day 7, as you can see here. And this replacement of the leukemia by the normal bone marrow, of course, will be accompanied by the loss of the PML-RER fusion in the bone marrow. Now, if we go to the PLZF-RER variants, we can see, in fact, that we have full differentiation. We transform an immature leukemia into granulocytic CML-like disease. But of course, as expected, from the absence of recovery of the normal bone marrow, this differentiated population is still entirely leukemic because they all harbor the PLZF-RER fusion. So this provided a provocative experiment that differentiation was not necessarily associated to disease clearance, and that it was likely that there would be two separate steps in the clinical response. And so this was the first problem with the differentiation therapy model. The second uncoupling was pharmacological, where again we used a number of synthetic retinoids represented here as NRX or acitretin, and we found out that those atypical retinoids shared the ability to induce differentiation, shared the ability to induce transcriptional activation of the targets, but failed to degrade PML-RER, which is not shown here. And as a result of the failure of degradation of PML-RER, clonogenic activity in methylcellulose was maintained, implying that in this system, we had differentiated the bulk of the leukemic cells, but we had not affected the leukemic stem cell phenotype, and that there was still a small proportion of leukemic cells which remain fully clonogenic despite reactivation of PML-RER, but in the absence of PML-RER degradation. So this was a second very strong evidence that transcriptional activation and differentiation was not the driver for response in APL. But the final experiment really came from very close, careful modeling in mouse models done by Julien Ablin, who was at the time a PhD, a very brilliant PhD candidate in the lab. And in a nutshell, using a variety of mouse models, what he was able to show is that transcriptional reactivation of target genes is responsible for differentiation. Degradation of PML-RER through the reformation of PML nuclear bodies and the activation of p53 was responsible for loss of self-renewal. And this, of course, was demonstrated also by the use of leukemia APLs, PML-RAR-driven APLs, in which the PML alleles was excised, or in which the P53 allele was excised. And in these variant leukemia, in mouse models, we keep the differentiation arm, but there was no loss of self-renewal, and therefore there was no therapeutic efficacy of the drug. And the same model actually holds true for arsenic. We have differentiation by passive reactivation of target genes through the disappearance of PML-RAR. The clearance of target promoters allows the reactivation of PML-RAR target gene passively. And the reformation of PML nuclear bodies, again, will activate P53 and allow loss of self-renewal. So there was a haunting question in the field, which was to know whether the two drugs would be synergistic or antagonistic. And taking differentiation as an endpoint, it was very clear that in cell lines or primary patient samples, as shown here, the differentiation activation of differentiation by retinoic acid was in fact antagonized by arsenic, with lower amounts, as you can see, of differentiation markers such as CD11B or CD11C. So for a while, clinicians kept the drugs apart from each other with the assumption that they were antagonistic for differentiation. However, exploration of mouse models showed the reverse. As summarized here, when we treat with arsenic or with retinoic acid, we somehow prolong survival, but we never cure the disease. This is accompanied by massive regression of the leukemia, as you can see here from in vivo imaging. But if we treat for only 3 weeks with retinoic acid and arsenic combination, we have a dramatic synergy for the elimination of the disease, despite the antagonism for differentiation. And this massive synergy for the elimination of the disease is accompanied by the cure of all mice. And so this was a very important experiment 20 years ago, which led to clinical trials that tested the association of retinoic acid and arsenic. The first trials were done in Shanghai. This is a result from the randomized clinical trial performed in Europe by the late Francesco Lococo. And as you can see, retinoic acid and arsenic in standard risk patients has a very, very favorable survival profile when compared to the historical retinoic acid plus chemotherapy treatment. I should stress that in contrast to kinase inhibitors, all patients get off treatment after 6 months. And the work performed in China by a number of different investigators have shown that oral therapy with arsenic sulfur combined to oral retinoic acid has exactly the same clinical impact, so that the disease can be essentially cured with 2 pills a day for 6 months or so. So this, of course, was a major achievement. I should say that after a couple of years, it became the gold standard treatment for APL all over the world. And this led us to the discovery that PML was the driver of response, which actually we did not know when we published the synergy between retinoic acid and arsenic. Well, this observation of the key role of PML in therapy response led us to rethink about the possible mechanisms. And I should say that Chen-Su and I had written a large number of reviews saying that the synergy between retinoic acid and arsenic was most likely mediated by a synergistic effect on PML-RAR degradation. Indeed, biochemically, the two degrons are different, so that retinoic acid and arsenic-induced PML-RAR degradation is synergistic through the combined activation of the two degradation pathways. However, I showed to you that the effector of therapy response is PML, and that arsenic directly acts on PML. And this can be shown here in these cells taken from mouse APL cells taken from 6 hours after in vivo treatment, where you see this beautiful reformation of PML nuclear bodies, even though the degradation of the PML-RER, which is in green here, is not yet complete. But despite this not incomplete degradation, we have this remarkable reformation of PML bodies. And this suggested that they could be, together with the degradation, a direct effect of arsenic on the normal PML allele, and that this enforcement of nuclear body reformation by arsenic activity on the normal PML allele could contribute to therapy response. This, again, was demonstrated when studying patients, and the study of these patients demonstrated that arsenic resistance could be linked to highly clustered mutation on the arsenic binding site of PML-RER, which is represented here. Arsenic binding residues in many protein are highly clustered cysteine residues. And here you can see that in this zinc finger, we have 3 cysteine residues. And in particular, arsenic is particularly avidly bound to adjacent cysteines. And there was a single D-cysteine residue in PML, which we proposed to be the binding site. And indeed, in artificial models, if we bind any of these 2 cysteines, we abolish arsenic binding. And we were delighted when a Chinese group a couple of months later reported in their arsenic-resistant patients very highly clustered mutations surrounding these 3 cysteines, as you can see here, which are therefore on-target mutations impairing arsenic binding and PML polymerization. I should stress that these were historical patients who had not benefited from the frontline arsenic-retinoic acid combination, probably explaining why they did relapse with arsenic single agent. However, what we reported a couple of months later in the New England Journal of Medicine was the same hotspot mutation. Actually, the most prevalent mutation here is this valine residue, which replaces the alanine residue. And the mutation in the patient whom we described was on the PML allele and not on the PML-RER allele. And in that patient, the PML-RER allele was not mutated, and therefore the occurrence of a mutation in the arsenic binding site of the normal PML allele in that patient established genetically the key role of PML binding by arsenic in therapy response. And so this led us to perhaps the ultimate model of APL treatment by retinoic acid and arsenic, wherein retinoic acid and arsenic will trigger independently PML-RER degradation by activating those 2 degrons. But at the same time, arsenic will bind to the normal PML allele and activate nuclear body reformation prior to the ultimate degradation of the PML protein. But for a couple of hours, we'll have massive PML nuclear body reformation, and this will activate P53, also probably trigger other pathways, including the E2F that was shown in some other experiments in fibroblasts. And these senescence markers then, these senescence effectors, P53 and E2F, will induce loss of self-renewal, at least in part through senescence, and direct the cure of APL. And naturally, this scheme now very naturally explains the clinical potency of arsenic, because in contrast to retinoic acid, which only acts on PML-RER and clinically is not sufficient to eradicate the disease, definitively, it only does it transiently, arsenic, via its dual activity to trigger PML-RER degradation and PML nuclear body reformation, will induce the full loss of self-renewal, which explains why, as I mentioned earlier, 70% of patients can be definitively cured by single agent arsenic. Quite a remarkable observation. So, how is P53 activated? Well, the answer actually is, for the moment, we don't know. What we know is that P53 can be recruited inside those PML, this PML shell. And this PML shell, work from my lab, which I will not discuss today, is envisioned really as a post-translational modification factory, at least in part through the recruitment of the key enzyme, the key E2 enzyme of SUMOylation, UBC9, and also its ARF cofactor. And all of the modifiers of P53, HOSP, HIP-K2, MDM2, ARF, DAX, you name it, you have it, they will all concentrate in PML nuclear bodies, at least transiently, together with P53. And so we see these stress-induced bodies, because not only arsenic, but actually any type of oxidative stress can induce the formation of these nuclear bodies. This will recruit P53 and all its modifier enzymes into these bodies where P53 will undergo a variety of post-translational modifications that will ensure its full activation before it exits the bodies and goes into chromatin. And indeed, in work that was published in GXMed a couple of years ago, we showed that PML is absolutely essential for oxidative stress-driven P53 activation in normal tissues, not in the leukemic context, but in normal tissues, and that PML is a key regulator of oxidative stress through its ability to undergo oxidative stress-induced polymerization and formation of the bodies and activation of P53, and in particular the antioxidant function of P53 in normal tissues. I also mentioned that PML controls E2F signaling, but this is not well understood mechanistically at the moment. And so in the last part of my talk, what I'd like to discuss with you is the logical conclusion from the APL story is that if PML nuclear bodies is the effector of therapy in APL, it's possible that it's also implicated in other kinds of malignancies. And we're here faced with a classic question is how can you drug a target? Are PML bodies druggable? And the answer is yes. And PML nuclear bodies are quite actually easily druggable through the ability of interferon to transcriptionally induce the expression of PML. PML is a massive transcriptional target of interferons type 1 or type 2. Actually, SUMOs are also induced by interferon. And if a cell that has a lot of PML is exposed to arsenic or to other forms of oxidative stress, you will induce the formation of PML nuclear bodies and recruit UBC9 and induce SUMOylation degradation of partner proteins, which is associated to senescence induction. And this can be visualized here in the mouse, looking at hepatocytes. And these hepatocytes have very few PML bodies in the normal conditions, but after treatment with an interferon inducer such as DIC and arsenic trioxide, you see this massive formation of PML nuclear bodies in these normal cells. So independently from the context of PML translocation, we have massive induction of formation of nuclear bodies, and therefore also massive induction, not shown here, of SUMOylation and degradation of partner proteins. So this sequence was actually explored in with my colleague and friend Ali Bazarbashi in adult T-cell leukemia, a disease prevalent in Japan, Africa, and the Caribbean due to infection with HTLV1. And what we're able to show is that induction of interferon and arsenic through this nuclear body formation can degrade some of the oncogene, one of the essential oncogenes of TAX, the HTLV1 encoded protein TAX. Well, we'll not discuss this today. This was extensively published. This has clinical efficacy in mouse models and to a certain extent in patients. And what I'd like to discuss is very recently published work in which in collaboration with a group in Institut Gustave Roussy in Paris and Hôpital Saint-Louis, we studied another disease, myeloproliferative neoplasm, and in particular those driven by the JAK2 activator mutation, which was discovered in France by William Van Schenker. And we know that this is a key driver oncogene for a variety of conditions, including polycythemia vera and primary myelofibrosis. So these, through the work of Jean-Jacques Quiladjian, also in Paris, it was shown that this disease is sensitive to interferon alpha, long-term administration. And when you look at the burden allele in these patients, after months of interferon treatment, you see a progressive decrease in the majority of patients, although the disease is not eradicated, but it's controlled in a large group of patients. And so what we proposed some years ago is that there may be a role of PML in interferon response in this disease. And if there was a role of PML in interferon response, it would be anticipated that they should be a synergistic effect of arsenic. And for this, I'll refer you to the publication for all of the data, but we mostly used a mouse model for the disease with a JAK2 knock-in allele with a GFP marker. We irradiate animals with normal bone marrow and leukemic bone marrow. So this is really an appropriate model for the human disease with a mutant clone and a normal clone coexisting. And we treat with interferon or interferon and arsenic and look at hematological response, molecular response, relapse after discontinuation and transplantation in mice. And to make a long story short, when we look at the molecular response, we have a molecular response to interferon. As you can see here, it's the blue line. So we have a decrease in the mutant JAK2 allele in red cells or in platelets or in granulocytes. But it's not complete despite 2 months of 2 months of interferon treatment. And if you add arsenic on top of interferon, you see that you have a much more profound decrease in the mutant and the mutant allele. And this is reflected in the fact that we can even eradicate the disease in some of the animals, and about half of the animals after treatment discontinuation, the disease doesn't come back, implying that we have targeted the leukemic stem cells in this model. And there is actually some evidence, which I will not present here, that we have an elimination of the leukemic stem cells, probably through a senescence-based mechanism. What I just want to emphasize today is that this is a PML-dependent effect, and to study the fact that it's PML-dependent, we generated myeloproliferative neoplasm with a JAK2 mutant knock-in allele on a PML null background, and did a competition study between PML null and PML proficient mice after interferon arsenic treatment. And what you can see is summarized here. This is the proportion of the mutant allele that is PML null. And as you can see, there is a remarkable enrichment from the original 50-50 equilibrium. You have a sharp progression of the PML null cells on all lineages, in the myeloid lineage, in the platelets, in the red blood cells. And this effect is not immediate, but manifested after 3 months of treatment, again, implying that the targeting of the leukemia-initiating cell by the interferon arsenic therapy implies, at least in part, PML. And actually, the involvement of PML is maximal in the myeloid lineage, on the platelet lineage. You don't have much effect of interferon alone in this setting. However, for the effect on the most obvious clinical manifestation of the disease on red blood cells, you already have an enrichment with interferon alone, implying that the effect of interferon alone is modestly mediated by PML, but the effect of the active interferon arsenic combination requires really PML for all lineages. So this demonstrates that arsenic sharply enhances interferon response and will clear disease-initiating cells, allowing the cure of some mice, and that the interferon arsenic response in JAK2 mutant leukemic cells involves a PML-driven senescence. And this was unexpected to me, that it was of such high amplitude, because it resembles very much the APL model, which suggests that PML-driven senescence could be a common effector pathway of therapy in different hematological malignancies, not only in APL, but maybe in a selected subset of leukemias. And the clinical feasibility and tolerance of the interferon-arsenic combination has already been demonstrated in ATL patients. It's not toxic. It's very well tolerated by the patients, so that this could drive clinical trials aimed at eradicating myeloproliferative neoplasm, which was something unachievable at the moment. So what I've tried to convince you in these two examples is to demonstrate the feasibility of back and forth oscillation between cancer patients and basic science. And I'd like to stress that some cancer cures can be mechanistically explained, and that the APL example stresses the key initial role of patients in the original empirical identification of retinoic and arsenic therapy, and the key role of in vivo study of physiopathology of response in mice in a way to establish the curative regimen. And so I'd like to argue that after enlightened trial and errors clinical approaches in cancer research, which have been done in the past 50 years, we should now promote rational combinations based on sound physiopathology. And I'd like to make a provocative statement, which we can maybe discuss in the question and answer. Do we really know how most common anti-cancer chemotherapy actually work? And I would say that I'm not sure that we really know how classic chemotherapy works in patients. And the natural correlate from this is, how can we optimize something that we don't understand? And of course, for acute myeloid leukemia, the classic anthracycline aracytosine combination, the so-called 3+5 combination, which is the classic treatment all over the world, I don't think we can improve it, because I don't think that we understand how it's active in these conditions. And so I think that physiopathological modeling of response is critically important also for novel targeted therapy, and required for rational combination of these targeted therapies. And finally, just a word to say that I think we need more basic and translational academia-driven science. We had it has led to therapeutic paradigm change for some leukemia, and I think that we collectively have an open boulevard for drug repurposing and rational combination through the combination of targeted therapy, classic therapy, immunotherapy, and this type of research driven by academia and not by industry, I think should be facilitated and financed. And in closing, I'd like to thank my collaborators and funding bodies. So I'm working partly in Hôpital Saint-Louis. Here, it's a 400-year-old hospital established out of the walls in Paris for the pandemics, notably the cholera pandemics. It was really out of the city at the time. It hasn't changed much, except that now it's in the middle of the city, and in Collège de France. And my work is supported by the Collège de France, the ERC, the SYNERGIZE, INSERM, University of Paris, Ligue contre le Cancer, Hôpital Saint-Louis. And I've mentioned my all-time collaborator, Valérie Lallemand, who really played a key role in these studies. And I'd be happy to take questions now. Thank you.
Thank you so much, Professor Thé. Very fascinating and illuminating talk, and especially this idea of how we can, drive the curiosity-driven research from the bench to the bedside. I would encourage the participants to put their questions in the Q&A chat box. I don't see any open questions thus far, but maybe I can start with a question myself. Just really enjoyed your talk, actually. So I was wondering, actually, especially with what you said about the interferon response, have you looked at how the other immune cells are changing? So in terms of immune phenotyping upon arsenic or retinoic acid, so what's happening to the T cells? Is there a TCR, BCR response? How does that change upon differentiation?
Well, I'm not an immunologist, but I have many immunologist friends. And they tell me it's impossible to cure cancer without the immune system. And I'm telling them, maybe you're right, but maybe you're wrong. In the mice models, you have to realize that you eliminate completely the disease in a week. Mm-hmm. And I'm unsure of any possible education, of the immune system in such a short time period.
Sure.
I'm not claiming it's the same in patients, because in patients, the eradication of the disease takes about 3 weeks. But in 3 weeks, you really get to 100% leukemic bone marrow, to no detectable leukemic cell at all. So this is something that we are studying slowly, because we're not immunologists. It's possible that there is a contribution. There were claims that in mouse models, the elimination of the disease, the eradication of the disease is not possible in deeply immunocompromised mice. But this was before the arsenic time. So I think it's an open question. As okay. Of course, there have been some recent publications that retinoic acid is an important mediator of the stromal to tumor interplay in some solid tumors, but this is not something that we have investigated ourselves.
Right. I guess I was also thinking more in terms of to promote differentiation, especially in the granulocytes, or in some cases there's some literature with neutrophils as well that maybe there's an innate response, not necessarily adaptive. I can understand to try to have an adaptive immune response would take much longer.
It's possible. It's possible. Yeah. We're not ruling anything out. And again, clinical observations is the key point.
Yeah, absolutely. All right, we have some questions coming in. Maybe I can read it to you. So from Weiliang Tam. Thanks, Prof. De Thé, for the tour de force demonstration of APL therapy, very inspirational. So what's the role of such differentiation therapy in solid tumors? And why aren't there many concrete successful examples in other cancer types?
Well, this is a very good question. Of course, historically, leukemia have been always much more simple than solid tumors. And that this is one of the reasons why many of us, who started in the leukemia field, did a little promenade in the solid tumor field, and then found it was too complicated and came back to leukemia. Probably one of the things is that there's some in APL in particular, APL can be considered as a monogenic disease. There are examples of patients for whom deep sequencing did not reveal anything else than PML-RER. It's not the most common frequent. Very commonly, you have FLT3 activation or other modest oncogene activation. But we have described some patients with PML-RER alone, period. So it's largely monogenic. And of course, if you kill the main driver, everything collapses. And if you activate PML bodies after you kill the main driver, it's even better. We all know that solid tumors are much more complicated and that they involve several sequential or not sequential activation or 4 to 6 different major oncogenic pathways. And so I think there are examples of differentiation in some solid tumors, but it's probably not enough. And I would argue, and actually we wrote a review in Nature Review some years ago on this, that differentiation in this case should not be the magic bullet. Differentiation can be a small contributor to the success. I think there's one good example for this, which is neuroblastoma. And as you may remember, neuroblastoma is treated by surgery, chemotherapy, and after that, it's given, retinoic acid is given as a differentiation therapy to fight an invisible disease, essentially, and it works.
Yeah.
It's not black and white, but it's clinically relevant. The relapse rate, if I recall well, go from 50% to 75%, from 50% to 25%. So in terms of patients, it's important, but it's coming when everything is done, when you already have surgery, chemotherapy, and so on. And my view of differentiation therapy, and I'm actually preparing a review for this now, is that I think we should, the model for differentiation therapy was APL for years. And so we all were expecting miracles. But I hope I convinced you that APL is not differentiation therapy. APL is senescence therapy with differentiation. And I think that the expectations of differentiation therapy were much too high. And I think differentiation therapy will work, but it will work as a small increment on chemotherapy, on targeted therapy, on surgery, just to increase and kill the last cancer stem cells.
Thank you for that. All right. So another question, what's your opinion about the role of PML in mitochondrial adaptation to oxidative stress, which could be induced by arsenic? And also this person would like to know your viewpoint about the mechanism of arsenic on P53 deficient AML.
Okay, so two different questions. I think the work from many groups has shown that there is a connection between PML and mitochondria, and we have not published, but we confirm this importance of PML on mitochondrial status. So very clearly in PML, in cells where you don't have PML, in cells where PML nuclear bodies are altered, you have a mitochondrial phenotype for sure, which is manifested at least in part by oxidative stress, basal oxidative stress. And this basal oxidative stress can be enhanced by arsenic. However, I think that our work showing mutations on PML in arsenic-treated patients demonstrate that the key target is arsenic binding to PML and not arsenic poisoning the mitochondria. It may occur, but I don't think it's the main driver for the eradication. I mean, the main driver is the master gene, and the master gene is PML-RER as the oncogene, and PML as the anti-oncogene. Now, concerning P53 deficient, so P53 deficient AMLs, I don't know, but P53 deficient APLs have been described. They're extremely rare. They're extremely rare, probably because it's easier to lose one PML allele than to mutate two P53 alleles. And so they're extremely rare, but they are resistant, unfortunately, to everything. They're chemo resistant, arsenic resistant, retinoic acid resistant, and all those patients that I've heard from have died, unfortunately. So we think that in APL, the mechanism of resistance is much more PML loss or PML mutation than P53 loss, even though P53 is a key player in the senescence and therapy response.
Thank you for that. All right, so have you looked at PML bodies in other subtypes of AML other than APL and JAK-mutated MPN?
Okay, so this is an interesting question. Sometimes, you should look at very old papers, and we published a paper 25 years ago on PML expression in solid tumors, showing that when you go from a normal epithelia to an early cancer, you have a massive induction of PML nuclear body formation, which we now interpret as oxidative stress. And then when the tumor progresses, you have a loss of PML. And this loss of PML could be interpreted now in retrospect as the loss of a senescence checkpoint. Work from several groups in Taiwan, in the US, in China, including that of Pierpaolo Pandolfi, has shown a number of PML degradation pathways which are activated upon tumor evolution. So in fact, a lot of solid tumors will activate, for instance, the casein kinase 2 was shown to degrade PML, and this seems to be important, if I recall well, in lung cancers. And so there is a relatively prototypical evolution in PML expression, with no PML expression in normal epithelial, high PML expression in pre-neoplastic and early neoplastic conditions, and loss of PML in evoluted advanced tumors. Now, with respect to leukemia, there is one setting in which there were PML abnormalities described, which is NPM1C mutant leukemia. This was described in two back-to-back blood papers 5 years ago by Bruno Fallini and Ali Bazarbashi. So that it's not as clear cut as APL, but there is definitely some defects in these conditions.
Sure. All right. Another question from Shara Kapinen. So I was wondering if arsenic effect, has it been tested in the non-JAK2 mutant MPN diseases? So those with CALR or MPL mutations?
Not yet, not to my knowledge.
All right. And perhaps the last question here from the attendees, Victor Ho. Do you have, so I might have missed this, but was there any effect of arsenic alone in the MPN model in the JAN paper? And do you have any opinion on innate agonists of RLR agonists or STING agonists as combinatorial treatment in cancer, maybe in PML or other leukemias?
Very interesting question. We have not done it, but I think some people should do it.
All right. Great.
If anyone in Singapore wants to do it, we're ready to collaborate or to help.
Right. Well, thank you so much. Any other questions from the panelists? If not, maybe I have another quick question, actually. I was wondering, it was not quite obvious to me. So in terms of the arsenic binding to PML versus the PML-RAR fusion, so
yes.
Is there a determination of specificity what binds to PML alone, or?
No, it's the same. It's the same binding site.
It's exactly the same. So,
It's
but
exactly
is it
okay?
So, but is the binding? Does the binding affinities change in the fusion protein versus PML?
No, this we don't really know. We don't really know. But you know, if we assume that the key role is to reform PML bodies, you can actually reform PML bodies even with PML-RAR. So there will be degradation, and that's what reason why in one of the images I showed and the dual combination with PML and PML-RAR, you can see full PML body reformation, even if you still have a little bit of PML-RAR remaining there. And actually with arsenic, some of that PML-RAR will be retargeted towards the bodies. So we really think that the reformation of the bodies is the driver of senescence and what is really required for therapy response.
All right. Well, thank you again for the very illuminating talks. I'm sure there are a number of other questions that perhaps people can reach out to you if they have. Sure. And yeah, thank you again for taking the time. Really, really enjoyed it. Thank you very much.
It's my great pleasure, and I hope that our next meeting will be not virtual, but will be over there. Exactly. We would love to have you here. Thank you so much. Thank you all. Thank you all for attending. Thank you. Bye-bye. Bye. Bye-bye.